Welcome to Going Around. So I'm going to keep this introduction really short. Most of you probably already know Chadi Abdallah. He's been a real fixture here for the past 5 years, but just a very brief background on Chadi for those of you that don't know him. You talk about human interest stories. Chadi has it. Chadi, really amazing story. Grew up during the Lebanese Civil War. Actually didn't get to graduate high school, which pretty amazing. Through the normal path. But after having an auto body shop store for some period of time, Chadi returned, graduated high school, through college, medical school at the Lebanese University of Beirut, really with no research experience at all. Came over, started his residency at SUNY Downstate. While, I don't know if people that know the residency, that's a tough residency. You're working hard. While doing that, still managed to get in labs, worked with people like Jeremy Copeland, then later with Sanjay Matthew, putting out papers, both primate studies, human, really just soaking up everything he could. And then we were lucky enough that to be able to attract Chadi here back in 2013, where he joined the Yale program. And really just amazing, truly one of the smartest people I know. Chadi could pick up anything and assimilate it into his already large knowledge base, whether it's technical skills, whether it's statistical analyses. His ability to just learn things is amazing. And Chadi also has this uncanny ability to look at a data set, whether it's an imaging data set, whether it's a clinical data set, whatever, and really pick the diamond out of the rough. I mean, he really has that unique ability. And that ability to take this large amount of data and to make sense of it is really quite amazing. So I'm not going to take any more time. I'm going to let you see for yourself. But I think today what Chadi's going to talk mainly about is his studies using ketamine, which I'm sure we hear plenty about here at Yale, but really as a tool to try to understand some of the neurobiology of the brain, some of the pathophysiology and mechanisms of drug action related both to psychosis and to mood, I think, and PTSD. All right, well, thank you. All right, Chadi.
All right, thank you, Jerry, for this introduction. And I promise I'm not going to talk about car unless we have a lot of time left over at the end of the presentation. So as Jerry mentioned that I started here as assistant professor in 2013. So I thought before telling you about what we've done over the 5 years, maybe it would be good to tell you what we would like to achieve over the next 25 years. So mainly what's our mission, what's our goal, and what's our plan of how we're going to achieve it. And here we see the photos of my son, George. So George, he's very helpful at home. So here he's helping in the backyard, collecting whoever's left paper on the grass. Here he's collecting the leaf in the fall, helping cutting the grass, do the weeding around the house in the spring, make sure our fridge is full of beer as if needed. And George also helped his mom. So this is Elena, my wife. She's endocrinologist. So George sometimes goes with her to the clinic. And I have this a bit of a fantasy slash phobia. So what if George will ask his mom, how does she treat her patient? So endocrinologist, it's going to be relatively simple. She'll tell George, we have patients, we check their blood sugar. If their blood sugar is high, we'll put them on a diet, we'll give them medication to reduce this blood sugar, and we'll see them regularly to make sure that the blood sugar is fine. But what about if George will ask his dad? I'm a psychiatrist, so what I'm going to answer him. So unfortunately, so far we don't have the blood sugar for psychopathology. And this would love to make it our goal. And we have this a bit of a fantasy that we can achieve it. And I'm going to tell you today how we would like to achieve it and why we think this is possible to do in the next 25 years. But before I do that, let me show you my daughter, Nai. So I cannot show you George without Nai. So that wouldn't be fair. So Nai here when she was 1 year old, and now she's 2 years old. So she's not Curious George yet, but she definitely has the moves. So back to our story. So if we think about kind of psychopathology over the past 100 years, a critical period is when DSM start being used. And we can think about DSM as psychopathology second generation. The problem with DSM that these diagnoses are based on behavior, and it has been really challenging for research purpose and for drug development purpose to find the underpinnings of these psychopathological disorder that we put them into kind of syndrome-like classification. And NIMH and other people involved thought maybe we have to have a kind of psychopathology third generation. And this is the RDoC approach. And the RDoC approach, most of you would be familiar with it, mainly trying to address some of the limitation of DSM. And definitely this has some strengths. So they try to use domain and construct instead of diagnosis with the hope that these domain and construct will better map to the biology. The main problem with RDoC that it's behavioral based. So RDoC remains behavioral based. These constructs and domain, they started from the behavior and attempt to go to biology. And this is, I think, it's a major problem, and it's very likely to bring us to the same issue that we've had with starting from depression to, for example, to biology. Depression is very heterogeneous. Clinical report could be affected by many, many things. So what about trying to go the other way? Is there a possibility to go from biology to behavior? And we think this is possible, and I'm going to present a model where this could happen. And this, we think that this is, instead of having a behavioral-based approach, we'll have a model-based approach for pathology. And I'll continue my envy of endocrinologists. It could, unconsciously, I could be competing with my wife, but consciously I'll make the case that, that diabetes could be a good example of pathology that's similar to something that we can observe in psychopathology, or something that we hope psychopathology will follow its lead. So with diabetes, you can have multiple insults. The good news for them that most of these insults are biological. And by themselves could be tested with biomarker. But you can have multiple insults that converge on a unique pathology, and would be the biological marker, the common pathway for a lot of these insults, and lead to many outcomes. It could be lead to cardiovascular problem, neuropathy, kidney problem, eye problem, and you can have a vicious cycle where the outcome become an insult itself and further exacerbate the blood sugar, which is the glycemia. And the critical part here, that glycemia, not only a common pathway, but it is causally related to the outcome. And this is what would be great if we can have similar model in psychiatry or in the study of psychopathology. We think that chronic stress pathology could be a good starting point. And for here, we're going to have a look at the potential insults that they will have when we're raising the question, do these potential insults have a common biological pathway? And if this common biological pathway is causally related to the outcome, and what could be that common biological pathway? So we can take an example, a trauma, cortisol, sustained threat, anxiety, depression. We know that behaviorally they can lead to these complications, some or all of these complications, like depression and anxiety, hyperarousal symptom in PTSD. And we know that there you have this vicious circle, so anxiety itself could lead back to depression. So if a person suffering from anxiety for a long time, this will create a chronic stress paradigm leading to chronic stress pathology in the brain, and ultimately perhaps this is what's leading to the depression. Or another example, you can have a person, cognitive errors, like a person suffering from schizophrenia, they have cognitive error leading to delusion, and these paranoid delusion will lead to insult, which is the sustained threat, and sustained threat will further exacerbate the chronic pathology in the brain. The main question here is not that these behaviorally related. We have a lot of data that these are behaviorally related. The question is how they are biologically related. Is there a common pathway? And is there a way to test it in a human? Also, just briefly about how these are related at the behavioral level. I think it doesn't take a lot of convincing for the crowd here to believe that chronic stress is bad. Chronic stress could be related to psychopathology. And here's a few number, even in mild stressors, like residency, for example. So this recent data show that the percent of depression prevalence, it could be up to 43% in resident physician. And more interestingly, that after starting residency, there could be a 16% increase in symptom of depression. So you can imagine in a mild stressor how this could affect psychopathology. The other example, if we look at the percent of male veterans who were exposed to high exposure of war in Vietnam. The PTSD prevalence is 36% in some studies versus 4% in the population at the time. And probably that's the most striking example is like refugees. Refugees, they could come from different background and unfortunately different suffering and different chronic stress level. But PTSD pathology in this population could go up to 86% in some really bad cases of refugee trauma. So you can see first the relationship between the stressor itself and the psychopathology outcome. And in some ways, all of us at some point, if we are stressed or traumatized enough, we are likely to develop psychopathology characteristic. And this relationship could theoretically be causal. So for example, some thought about early life stress, how it could cause psychopathology later in life, or it could be a trigger like the case of PTSD. We may have certain predisposition or certain resilience factor that protect us from PTSD, but there is a traumatic event that started all at some point. And it could be an outcome, like I mentioned, about anxiety and paranoia, how they could themselves to chronic stress. So in that sense, is really in the majority, if not all, the psychiatric disorder that we do have a huge component in chronic stress. And if this chronic stress is negatively affecting the brain, and what we're calling here brain chronic stress pathology, then perhaps this common across many psychiatric disorder, and this could also raise the question why we have a similar biological finding across disorder. We can see a reduced hippocampal volume in depression. We show it in generalized anxiety disorder. People show it in PTSD. People see it in schizophrenia. Is there a biological common pathway across these disorders? And this common pathway, is it causally related to chronic stress as an outcome and also as predictor of complication? So these are important questions. And other questions could be antidepressants. Antidepressants, we use them for anxiety, we use them for depression, we use them for PTSD. And is there a common pathway that's how it's beneficial for multiple disorders? So in this circle, we're going to mainly focus on this brain chronic stress pathology, develop this as the biomarker, and advance the model, and then go back to the behavior. So for this, we're going to use some of the preclinical finding, and the main thing in this model, which is kind of amino acid-based model for chronic stress pathology, is here what we see as an example from pictures of dendrites. So in the top right here, we see the control dendrites from the control animal, and here we see the stress animal. This is relatively mild stress of chronic unpredictable stressors. And the changes that we observe, and this is repeatedly presented in the literature, is you have this reduction of branching or shrinkage of the length of the dendrites, or you have reduction of the number of spines where the synapses occur. And this is mostly, I'm talking here about the prefrontal cortex and some of the similar changes occurring in the hippocampus as well. But the main outcome in this model is that you have this synaptic disconnectivity. So these pathological changes converge on this synaptic disconnectivity in the prefrontal cortex, which is the area I'm going to focus on it today. And this seems to be directly related to pathology in the animal literature. And there are some hypotheses at what could be leading this. So for example, it could be disruption in the glutamate neurotransmission or reduction in glutamate uptake by astrocytes. We may agree or disagree, but the common ground is here, the synaptic disconnectivity in the prefrontal cortex. And kind of to get people on the same page, I'm going to highlight a few processes in the glutamatergic synapse that's relevant to the talk today. So in a typical glutamatergic synapse, we'll have glutamate release in the synapses. The glutamate release in the synapse will activate postsynaptic receptors, so mainly relevant to the talk today would be AMPA and NMDA receptors. And this glutamate release will be taken up rapidly by astrocytes, which is the kind of accessory or helpful cells, but they do a lot of work in regulating glutamate neurotransmission. So glutamate is taken up by astrocyte, converted to glutamine, and glutamine is shuttled back to glutamate. And this would constitute the glutamate-glutamine cycle. And under normal condition, the glutamate cycling will lead to postsynaptic activation. And so, for example, if you increase the glutamate release, you'll increase postsynaptic activation. One feature here that these synapses are have good plasticity in a sense that a synapse can increase the number of postsynaptic receptor, for example, AMPA or NMDA, and this will increase the strength of the synapse. So the synapse will become more stronger. And what we mean by more stronger, that's for the same amount of a glutamate released, you have increased probability of activation postsynaptically. So the things to remember about the synapse, that we have a glutamate-glutamine cycle, which is release of a glutamate in the synapse, taken up by astrocyte, converted to glutamine, and send it back, and this reflects the glutamate release. And we have postsynaptic activation, and this depends on the strength of the synapse, which is, to simplify, the amount of receptors that you have postsynaptically. So in this model, you can kind of speculate that perhaps in a human we have something similar. When people are euthymic, where their mood is fine, they're not suffering from trauma or prolonged chronic stress, they have the right ingredient. They have the right amount of presynaptic glutamate to release, the right amount of postsynaptic AMPA and NMDA receptor to activate postsynaptic activation, and you have enough of these synapses. In fact, you have a lot of these synapses, perhaps more than what you need. What happens when the animal is chronically stressed, or to speculate about a human, when a person, for example, is depressed and suffering from depression for a long time, or trauma? What happened, we think, that first you'll have reduction of the number of these synapses, so the brain regions and the prefrontal cortex doesn't have as much synapse as it has before. And the second critical part, that the remaining synapses, they're not as strong as before. So they have little remaining postsynaptic receptor to activate. And the hope, and at least in the preclinical literature, it seems antidepressants, especially ketamine, seem to reverse this synaptic abnormality. This synaptic connectivity that we believe is critical to chronic stress pathology seems to reverse after treatment with antidepressants. And we will talk a little bit about potential mechanism, how's that happening. So going back to our model that we would like to discover, what's the blood sugar of CSP, chronic stress pathology. So you probably guessed it by now. We think that synaptic connectivity could be critical. It perhaps, this could be the biomarker that would parallel blood sugar for psychopathology. The question is, it's not easy to test it in human. Otherwise, we don't need 25 years to answer this question or to reject it. The problem is we don't have the tools currently to reliably and consistently in a reproducible manner to measure synaptic connectivity. But we have other tools that indirectly target this biomarker, and today I'm going to present 2 examples. So before I start telling you about the data that we have, I thought it would be good to follow Elon Musk's approach. Let's have a secret master plan that you all will promise me you're not going to share it with nobody. And for Tesla, they had a 4-item secret master plan. So what's our lab secret master plan? So the first secret master plan, the first item is to work to have chronic stress pathology acknowledged as a disease. And this is really critical because without this acknowledgement, every time we have a finding with the chronic stress, we're going to attempt to fit it into depression, we're going to attempt to fit it into PTSD, we're going to attempt to fit it into schizophrenia, and this is really problematic for the progress of the research. The other issue that is equally important, if not more important, is that you're not going to get money or you're not going to get paper published unless this is a real problem. Unless we show that it is a real problem and it's relevant to treating psychopathology. So NIH funding will be related to acknowledging something is a disease or not. And taking an example, PTSD, I was surprised to know that it took more than 60 years to move from the definition of shell shock to including PTSD in DSM, I think DSM-3. But now with PTSD acknowledged as a disease, there is a lot of resources and there are a lot of bright minds working on solving this issue. So I think it's critical to kind of work on this, helping identifying chronic stress pathology, definition of it, and accepting it as a disease entity. And to do this, we have talks, like I spent 10 or 15 minutes of my talk to preach you about it. But other things, we have collaboration. We can promote publication. For example, John Crystal, Nat Averill, and I established a chronic stress journal last year. And I'll take this opportunity to, next time you have a relevant paper, I will encourage you to strongly consider chronic stress. Not only you will disseminate your finding in an excellent journal, as the editor said, but also you'll support the cause and unconsciously you will contribute to our secret master plan. So chronic stress is trying to focus on the prevention, identification, and treatment of chronic stress pathology, and any relevant literature would be welcomed. So the second item is biomarker testing and optimization. A lot of the studies that we read and we do are really knowledge-based, not much biomarker-based. So what we're interested into the biomarker that we use, what does it tell us about the brain? And this is a good point because first we're biased because that will help increase the impact factor of our papers as well as the likelihood of our proposal. But really there is little work about reproducibility of these biomarkers. And unfortunately, many of these biomarkers, they don't do very well in replication. So what we're trying to do is revisit a lot of these biomarkers and optimize them with an eye on reproducibility versus knowledge. We're still going to get knowledge, but we're going to prioritize reproducibility. And to give an example, so for example, if a patient population has a lot of movement in fMRI, and if a GSR is the only way to address this movement, I'm saying if, I'm not saying the answer, but if that's the way to go, even though GSR will make the interpretation much more difficult, we're going to prioritize reproducibility over interpretation. And we're going to do through a number of biomarkers that I'm not going to present today. And the third item is pattern analysis. We know that, for example, hippocampal reduction seems to become in a lot of psychiatric disorder, and it's difficult to replicate even within one psychiatric disorder. So you can randomly select 60 depressed subjects and another random 60. One of them may have reduction on average of hippocampal volume, the other will not have it. So what about having kind of, instead of looking at unique variable, what about looking at pattern analysis, looking at two variable or two multimodal approaches, mainly including multivariates in our development of the biomarker. And we hope to do this by using two approaches. The first one is a model-driven approach. Mainly based on a priori hypothesis, we can predict how these patterns should change, and or based on different knowledge. For example, here I put example network topology. Based on our knowledge of graph theories and topology of networks, we can predict we can summarize the pattern of changes in each brain region based on these. And then this will use it as a biomarker. The other approach is to have a data-driven approach. Mainly we'll take all the variables that are available. So for example, fMRI will generate 91,000 voxel time series. So one approach would be to take this 91,000, but hopefully we'll do well with the model-driven approach to have a feature reduction from 91,000. We think we're working right now on 300 or something close to 400 features. And then use a data-driven approach, example, machine learning algorithm, to identify the patterns that's a good biomarker. And the problem with machine learning that it requires a lot of data. And unfortunately, in the clinical literature, Mostly we have small studies where you have 60 subjects. If it's too large study, will be in the hundreds. And this is not going to do very well for machine learning. So our approach for this is to use a variable that are relevant to the biomarker of synaptic plasticity. And still, we have a lot of data, and we're using, as a first step, we're using age. So for example, so far, we compiled something between 15,000 and 20,000 scan, where the focus is going to be on age. And the similarity between the changes with aging and the changes following chronic stress pathology will help us figure out how to go back to the clinic. And the last item, which is going to be the focus of my talk today, is to focus on interventional approach. And this interventional approach is mainly to, instead of having cross-sectional analyses, and which are very problematic for reproducibility, what if we start by having mechanistic interventional development of these biomarkers? And when we trust the biomarker and we're fully confident the biomarker is answering our question, then we'll go back to test it in the clinic. And here where we're going to, ketamine is really a powerful tool to use it. So if you remember this slide that I just presented, that we have this reversal of synaptic disconnectivity after treatment. The good news that ketamine in animals does that within 24 hours. So you have reversal of synaptic disconnectivity within a 24-hour of treatment in the animals. In the human, you have, if people were depressed, we have approximately 50% of them will achieve response, also 24-hour post-treatment. So this is really offering us a really powerful tool to study these biomarkers. So what do we think about what are the mechanisms of action of ketamine and other rapid-acting antidepressants? So to keep it simple, I'm going to highlight two processes that seems to be important and that we're going to talk about today. The first one, it seems that glutamate, following acute administration of ketamine, you have a glutamate surge. So mainly increase of glutamate release following administration of ketamine. At least this was demonstrated in animal, and we have indirect human evidence to support it. And we're going to present today a bit more direct evidence for this ketamine-induced glutamate surge, acute glutamate surge. The other critical part, and that seems also relevant to antidepressant effect, is that 24-hour post-treatment, you have this synaptic changes, mainly overall increased spine density, which then increase the number of synapses, as well as increasing the strength of these synapses. So we have two glutamate neurotransmission changes in the prefrontal cortex. One acutely, which is during infusion if it's a human study, and one 24-hour later. So the first biomarker that we're going to use is based on carbon-13 magnetic resonance spectroscopy. And how this method works, mainly we'll have a patient, will place IV, and will infuse labeled, for example here, labeled glucose, labeled with carbon-13. And the glucose is the main fuel of the brain, especially of the neurons. And the majority of energetic needs in the brain are to maintain this glutamate neurotransmission that I just described. So glucose metabolite will enter the Krebs cycle for oxidative energy production. And after that, they will label a glutamate. So what we do, we measure the concentration or the enrichment of glutamate and glutamine every 5 minutes over 125 minutes. So first, glutamate is enriched with carbon-13, and then glutamate is released in the synapse, taken up by astrocyte and converted to glutamine. And now glutamine is enriched as well. So we can measure it here, and we can look over time. So this is the blue one, is an example of a time course for glutamate carbon-13 enrichment. And it will see that it over time, early time point, it will go rapidly up, and then it will reach a steady state. And this thing, based on preclinical literature, show that this is reflect energy production, oxidative energy production. And as we said, because oxidative energy production needs are mostly to maintain postsynaptic neuronal activation. So pretty much this is an indirect measure of postsynaptic glutamatergic neuronal activation. And the second one is we can look at glutamine enrichment, and we see we have similar time course that over time, the more glutamate is released in the synapse and taken up by astrocyte converted to glutamine, the more you can estimate this cycling. So here we can try to guess two measures. The first measure is energy production, and we believe this related to postsynaptic activation, and there are good amount of preclinical data to support that. And the second measure is the glutamine cycling. So this is a bit of a powerful method that in vivo, in human, you can get estimate at the level of estimate unique to glutamatergic synapses. And the first study was done by Jerry Sanacora and others, and in this study, we had 17 healthy subjects and 23 patients with major depressive disorder. At the time, we could only get the spectroscopy in the occipital cortex, so the data are from the occipital cortex. So the first finding was that depressed patients, they have reduction in energy production, about 20-something% reduction in energy production. And this is not surprising because we just said that synaptic strength is reduced, and also the number of synapses are reduced in depression. So this is not too much surprising. But the surprising part is this one, that if we look at the glutamate cycling in this population, again, in their occipital cortex, we see that the glutamate cycling was maintained. So glutamate cycling was not affected, but you have a reduction of energy production. So what could be explain this, and why is that important? The first thing here, we look at the figure to the left. So this is average of preclinical data, which is are in red. And this is the average of each study, not of one individual. And this is the average in a human study. And what we see here, we see a nice one-to-one correlation. This energy per cycle is very stable across the species, across mood states, so here, or mental states, so here some of the animals will go to sleep or will have complete anesthesia, and this energy per cycle go up together and go down together. And what we have in our data, if we look at the healthy subject, we have something similar, exactly almost the same. You have, of those 17 subjects, 8 to 9 who are above the predicted line, and 8 to 9 below the predicted line. And if you get the mean is exactly on the blue line. So this will fit nicely with the other normal data. If we look at the depressed subject, here where you will see the difference. Of 23 depressed subjects, we only have 2 subjects above the predicted line. So we have this robust reduction of energy per cycle. And what could explain this? We think that this will be related to preclinical data showing that chronic stress pathology reduce synaptic strengths, and reduce that by reducing AMPA receptors and reducing NMDA receptor. And there is postmortem data actually to show that even major depressive disorder, in postmortem data, they do have reduction in an NMDA receptor. So our interpretation for this, one of the interpretation, if we rule out other methodological limitation that we're trying to solve, and I'm going to show some of that data, the neurobiological interpretation would be that we're capturing reduction of energy per cycle. So to go to prefrontal cortex. So in the prefrontal cortex, now we have the methods that we can do it in the prefrontal cortex, and this is example of preliminary data where we have, we can get the average on this brain region and do the same, get the glutamate enrichment, glutamine enrichment, get the time courses. The difference here, we infuse acetate. What I didn't mention that if we infuse acetate, acetate is taken up by astrocyte exclusively. It doesn't go into neuron, and this is a very important tool to look at the astrocyte deficit. If there is glial deficit in depression, we will capture it by giving acetate and show that there you have reduction in glutamate metabolism in these astrocytes. So preclinical data also from Jerry Senacora's lab, where they looked in the prefrontal cortex of animal, you see that chronic unpredictable stress reduce enrichment in the brain if you give acetate. And here our preliminary data from 10 depressed subjects and 5 healthy. And we see similar patterns. So you have reduction in glutamate enrichment and reduction in glutamine enrichment in the depressed subject. If we look, so almost half of these depressed subjects, they are on SSRIs or SRI, and half they are non-medicated. We see that for this, the measures that we think are relevant to astrocyte deficits, those medicated, they continue to have actually a reduction, if not even more than those who are not medicated. The other thing that we can look is at energy per cycle. So from infusing acetate, we can get estimate of energy per cycle. And here on the left, reminder of the occipital data where we have a reduction of energy per cycle compared to healthy control. And here the preliminary data that we have in human. We see the same pattern of reduction of energy per cycle compared to the healthy control in the prefrontal cortex. And it seems that treatment and with antidepressant perhaps may be increasing it, perhaps not. We'll have to wait and see the full study. So we now get funded with R1 to do the full study, and we're going to test it. But if these preliminary data will hold in the full data set, this will tell us that perhaps right now we have a good biomarker not only to capture the depression-related pathology, but also the effect of antidepressant. So getting to ketamine. For ketamine, we said that ketamine will lead to a glutamate surge. And here there are two possibilities for the effect of ketamine on cycling and on energy production. The first possibility that ketamine will increase cycling, and this is very highly expected. And then because cycling and post-energetic needs are coupled, so then it will increase cycling and it will increase as well as energy production. The second possibility, because ketamine is an NMDA antagonist, there is this possibility that perhaps ketamine, in addition to increasing glutamate release, is actually reducing synaptic strength by blocking postsynaptic NMDA receptor. And these are the data from animal studies. So we'll see here on the left, we see data from rodent. This was intraperitoneal injection. So this is really relatively post-ketamine, not during ketamine, but we're not sure this post-ketamine, how much it will map to the IV ketamine in a human, or if you have more kind of delayed effect. But if we look at this, the first time point, we're pretty sure that this is the closest it gets to during infusion, because this is acutely after you give the intraperitoneal ketamine, so you do have the ketamine is still available. And what you observe here, so the green is the glutamine enrichment, and as we said, this is mostly will reflect glutamate cycling, and the blue is the glutamate enrichment, and this will reflect energy production. And here in the first 10 minutes, we see a robust increase in the glutamate cycling, but no increase in energy production. And the second step, which is 10 to 20 minutes later, we see increase in both. We have increase in glutamine and increase in glutamate, although glutamine remain relatively slightly different than glutamine. And then after a few minutes, perhaps at this time, ketamine is getting out of the system, you have reduction in both, so you don't have significant increase. One thing about ketamine, at least in human, that if we are infusing ketamine and post-ketamine injection, the first 10 minutes, patient will suffer from psychotomimetic effect. But 10 minutes later, they will not have psychotomimetic effect. So we're not sure how much this preclinical data will map to the behavioral effect in a human, but if they were to map time-wise, that would be the time point where the subject is experiencing psychotomimetic effect, and that would be the time point where they are not experiencing psychotomimetic effect if this was human data. So what about the human data? So we use the same methods in the prefrontal cortex. We measure glutamate and glutamine. We have healthy subject, we have depressed subject, who are one day they will get carbon-13 with normal saline, and one day they will get carbon-13 with ketamine. But here we have ketamine, we're measuring the effect of ketamine during infusion, not post-infusion. And these are the results. So we see that first, the red is the glutamine enrichment, which reflects glutamate cycling. We see about 13% significant increase in the glutamine enrichment, providing perhaps the first human evidence that ketamine increased glutamate, induced glutamate surge in human, and you have a biomarker to measure it. The second one is that there is no changes in glutamate enrichment, which we think reflects energetic need. And this is a bit of our surprise, because a priori we would have thought that what we're observing here, 10 to 20 minutes, will map to the human. And what we're seeing is actually what we're observing in the first 10-minute post-infusion is what's mapping to the human data. And this could be several possibilities. One possibility that maybe we don't have the sensitivity for the small increase in the glutamate changes. Maybe the method, keep in mind that the preclinical data is 11 Tesla ex vivo. The human data is 4 Tesla in vivo. So you have a huge difference in sensitivity. So perhaps we're still observing the same increase, 10% and 30%, but we don't have the sensitivity to capture the glutamate changes. And the other possibility, which I would believe it's more important and interesting to study, is that the first 10 minutes after ketamine infusion is the time when you have the psychotomimetic effect, and is the time where you have increase of cycling without increase in energy production. So that's the time of reduced energy per cycle. And what makes this interesting, that's here first on the left, if we looked at the healthy subject and MDD subject, the changes were relatively the same. Mainly glutamate increase and no changes, 1 to 2% changes in glutamate that are not significant. But if we look at the correlation with psychotomimetic effect, in the top right we see the CADS measure of dissociation. We see that the glutamate over glutamine enrichment is the one that predicts psychotomimetic effect. And this, in this sense, if we were to interpret by speculation, if we were to speculate, we'll speculate that subjects who have reduced energy per cycle are the ones that suffer the most from psychotomimetic effect during the infusion of ketamine. And this perhaps very relevant to schizophrenia literature. Because of schizophrenia, most of the literature so far indicate that this ketamine-induced surge is underlie or is causally related to the psychotomimetic effect of ketamine. Here, it's raising the possibility it's not actually the increase of glutamate cycling, it's the increase of glutamate cycling while blocking postsynaptic activation, mainly the mismatch between input information and output signaling. This perhaps what's causing the psychotomimetic effect. And the hypothesis would be that if after 10 to 20 minutes, like if this is a human data, 10 to 20 minutes after infusion, there will be no psychotomimetic effect. In this 10 to 25 minutes, you continue to have increase in cycling, and perhaps you start to have increase of postsynaptic activation. And this would be the relevant for depression. Because for depression, as we said, the model, you need to activate postsynaptic receptor to induce synaptic plasticity that will be optimized by 24-hour post-treatment. So we think this preliminary data is pretty exciting. First, it is preliminary data because these are only 14 healthy subjects. So we hope NIH will support us to do the full study where we'll do a large sample, confirm the reproducibility, and confirm that in a human, you have something similar to what you observe in animal, that during infusion, you block postsynaptic activation while increasing presynaptic glutamate release, and that 10 minutes after infusion, when you don't have psychotomimetic effect, you actually activate both, increase cycling and increase energy production. And this could provide a biomarker that on the one hand can be used for development of psychotic targets, antipsychotic treatment, and on the other hand can be used for rapid active antidepressant. So how are we with time? Here I have 43 minutes, so I guess I have 15 more minutes. So in these 15 minutes, I'm going to try to focus on a different biological measures. And this biomarker is based on networks instead of based on synapses. So this measure is relatively simple. We do fMRI, functional fMRI, and we'll look at the functional connectivity between the brain region and the rest of the brain. And the average of this connectivity of a brain region with the rest of the brain is called global brain connectivity. So to simplify it, if I'm measuring global brain connectivity for the prefrontal cortex, high global brain connectivity in the prefrontal cortex will tell me that the prefrontal cortex is highly connected to the rest of the brain. Low global brain connectivity in the prefrontal cortex will tell me that the prefrontal cortex is disconnected from the rest of the brain. So that's a simplified measure. So there were a number of studies by Naomi and Alan and John where they have in a ketamine during infusion, they show increase of GBC, global brain connectivity during infusion. So that's interesting. The second interesting observation that Alan did a number of studies in bipolar depression, in OCD, and also in schizophrenia, where he noticed you have a reduction of a GBC in the prefrontal cortex. So putting this together, we thought, maybe this GBC is reflecting the synaptic disconnectivity that we hypothesize is going to increase during ketamine, and it's going to reduce in a psychiatric disorder that they have a huge component of chronic stress. So let's test that. So the first study we did in collaboration with James Morrow at Mount Sinai and help from Alan here at Yale. We looked at 57 depressed subjects versus 25 healthy control, and we proved this support our hypothesis that you have widespread reduction of a GBC, which is in blue here on the left, in the prefrontal cortex. And on the right is showing to which intrinsic network these connectivity deficit will map. The second question is, okay, so if this is related to depression and related to synaptic connectivity, maybe ketamine can increase it because that's what ketamine does in preclinical literature. So in a smaller sample of 18 treatment-resistant depressed versus healthy control, we can show that before treatment, you have this blue where you have reduction in prefrontal global brain connectivity, and after treatment, what happened? You have normalization. You have almost complete normalization of the reduction in the prefrontal cortex with the exception of this small cluster in the ventral prefrontal cortex. If we look at the data differently, we look here, the red spread is the number of voxel that are abnormal before treatment, and blue is the number of voxel that are abnormal 24 hours post-treatment. We see a remarkable reduction from 2,100 voxel to something 310 voxels. And if we focus on the prefrontal cortex and we look at the spread of the global brain connectivity in the prefrontal cortex. So the red distribution here is the distribution in healthy subject in the prefrontal cortex, and the blue is the distribution in patients suffering from major depressive disorder. And we see on average, you have a left shift. That's on average, the prefrontal cortex, you have a significant reduction in the global brain connectivity, indicating that in depressed patient, somehow the prefrontal cortex is disconnected from the rest of the brain. So what happened after ketamine treatment? So out of these 18 subjects, about 50% of them responded to ketamine. And if we look at the responder, we see nice overlap. So the red remained the same healthy subject, and the blue are those who responded to treatment. You see a remarkable overlap after treatment, indicating normalization. What about those who did not respond to treatment? So those who did not respond to treatment, they continue to have the same problem, which is disconnectivity between the prefrontal cortex and the rest of the brain, and we see similar distribution, similar effect size to what you see before treatment. Let's see how much time we have. So for the sake of time, I'm going to skip this part, and I'm going to tell you what were the results. So mainly we had a suspicion that can we provide evidence that these GBC changes are directly linked to glutamate in neurotransmission? And we did a number of studies, and this is the conclusion of this study, that they are perhaps related. So we can give lamotrigine as a glutamate release inhibitor. We show that lamotrigine will reduce prefrontal cortex connectivity, the blue. We can give ketamine. Ketamine will increase global brain connectivity, and we now know that ketamine increases also glutamate cycling. We can give a placebo and show the biomarker is very stable, so it stays close to zero when you give normal saline, does not change. You give them ketamine, you increase it. You give lamotrigine, which is a glutamate release inhibitor, before ketamine, and you reduce the ketamine-induced surge. So coming to the final study that I'm going to present today is, so now we know that we have a glutamate surge in depressed in healthy. We know that we have surge in global brain connectivity in the prefrontal cortex in healthy. What about in depressed subject? Do we have the same surge? And the other things that we didn't answer before, that most of the study that we've done with ketamine in depressed, they were not controlled. They were open-label ketamine. So we're looking at pre-post. We don't know how it's going to do compared to placebo. And the second question, is this GBC surge during infusion related to the antidepressant effect 24-hour post-treatment? And here are some of the answers. I believe we answered the first question, and we have preliminary data about the second question, which is the relationship between GBC and improvement. So first question here, we see again that the biomarker is doing excellently well in placebo, close to zero with not much changes during infusion, good test, retest within session. We have ketamine significantly increasing GBC, and here we had an additional NMDA receptor, which is lanicimene 100 milligram. And lanicimene, although numerically it increased it, but it was not significant. Sorry, I see it. You have to believe me. Okay, so then I'll repeat. So we have placebo increase. We have placebo, good test, retest stability, no changes. Ketamine increasing prefrontal GBC, and lanicimene 100 milligram is not changing prefrontal GBC. But numerically, it's slightly higher, but not significantly different or significantly different to placebo. What about 24-hour post-treatment? Again, we have good test, retest, reliability, no changes in the placebo group. In the ketamine group, we have significant increases, and these increases are significantly higher than changes in placebo. In lanicimene, here it gets a little bit interesting. So although lanicimene did not significantly increase GBC compared to placebo, but if you look at the pre-post changes, there are some significant changes. So it kind of suggests that 24-hour post-treatment lanicimene, 100 milligram, it seems to start to pick up, but not sufficiently to increase GBC compared to placebo. So what about the relationship between the glutamate surge during infusion and the GBC surge during infusion and antidepressant effect 24-hour post-treatment? And here we see the correlation between the GBC surge during infusion in the prefrontal cortex and improvement 24 hours later in the placebo group. So there is absolutely no significant relationship. In the subject who got the active drug, we see a significant positive correlation. So the red, again, are the ketamine, and the blue are the lanicimene. We see that the amount of increased, the higher the increase of GBC during infusion, the better the improvement 24-hour treatment. Kind of telling us that perhaps the problem with lanicimene that it didn't do well during infusion to predict response at 24 hours, but it's still, the amount that it increased is still predictive, raising the question whether the problem with lanicimene is to just use different dose, different type of administration, or kind of optimization of administration. So to summarize, I told you a story about how we can potentially use a model of psychopathology for fourth generation psychopathology. Another story about secret master plan that you all promised not to share with others. And I also talked about two unique biomarkers that we think they have a lot of promise for the study of chronic stress pathology and other related disorder. And I would like to thank the many investigators and colleagues and collaborators to help make this work possible. I'll thank Nat Averill, John Crystal, Stephen Southwick. And about Steve, yesterday he has a very important advice to me. He said, be mindful and have fun. So definitely I had fun. I'm not sure about the mindful part, but thank you, Steve. And also I would like to thank Jerry. A lot of the study that we did was mentored by Jerry and supervised, and many of the study he ran them. And Graham and Doc for their help and supervision. And Sanjay, Matthew, and Ramiro Salas from Baylor that they shared their data with us for ketamine fMRI. The same with James Murrow from Mount Sinai. And then Mathalone, who shared the data with the lamotrigine fMRI with us to do these studies. And Bill Deacon, who shared the data with the lanicimene ketamine to do these studies. I'd like to thank all the institution and the funding agency that they helped us make this work possible. And a special thank to the A-team, A-teamers. So if you think that this picture was taken from ugly sweater party, you got to try it. And I'll tell you who was the winner. So the winner was Tim. He's hiding in this picture, but he's really had very artistic ugly sweater. So he was the clear winner. But here we didn't have a clear winner. So Tim was the clear winner for the ugly sweater competition, but for the smile competition, we really didn't have a main winner. We had big smiles, small smiles, but all smiles were special. So. And I also would like to thank the A-teamers who helped us and worked with us over the past 5 years, and they now moved to other adventures, so they're not with us anymore, but would like to thank them. And finally, if you were wondering what's this bird in the top right of the slides, what is it doing? So this is mainly the logo of our Emerge research program, and the logo and the name of our program reflects our hope for patients suffering from mental illness, that they may emerge from their suffering like the phoenix rise from the ashes. And thank you.